Hello and welcome to HIV RNA Test Guide Podcast, your trusted source for HIV testing, with over 4,500 plus testing labs across the United States. For more information about STD and HIV testing, check the link in the description or in bio section. Today, we're answering a question that so many people are asking. When will the FDA approve an HIV cure? It's exciting to hear all the news about breakthroughs in HIV vaccines and potential cure therapies. But there's a big difference between a promising discovery and a real approved cure. So, will the FDA actually approve an HIV cure in the near future? In this video, we'll break down the science in simple, easy-to-understand language, explain where the research truly stands today, and share practical steps you can take right now to stay informed and protect your health. So don't go anywhere. Stay tuned and listen closely. Today's podcast is packed with valuable insights.
Hello and welcome to HIV RNA Test Guide Podcast, your trusted source for HIV testing, with over 4,500 plus testing labs across the United States. You know, if you've been keeping an eye on medical news, maybe over the last decade or so, you've definitely seen those headlines popping up. HIV cure breakthrough imminent.
Yeah, those grab your attention for sure.
They do. But the question we get constantly, like almost weekly in our inbox, is much more down to earth. When is the FDA actually going to approve an HIV cure therapy?
Right.
So today, we're going to try and provide that realistic timeline, sort of cut through the hype, the hopeful headlines, and really focus on the science and, importantly, the regulatory side of things.
That's such a critical distinction to make, because the jump from, say, a promising result in a small trial, a handful of patients, to getting a therapy approved, manufactured, distributed globally, it's a huge leap.
Monumental.
Yeah. So our job today is really to bridge that gap for you based on, the current standards the FDA uses for safety, for efficacy. What does it really take?
Okay, so before we dive into the actual science, we need to get our terms straight. Because cure, well, it actually means a couple of different things to researchers, doesn't it?
It absolutely does. So when scientists or even the media talk about an HIV cure, they're usually talking about one of two things. First, there's the sterilizing cure.
That sounds like the ultimate goal.
It is. That means complete removal. Every single virus particle gone from the body. No HIV left anywhere. That's the gold standard.
Okay. And the other one?
The other one is what we call a functional cure.
All right. Functional cure. Now, if I'm listening, I might hear that and think, well, isn't that just really, really good treatment? Like, how is it a cure if the virus is still technically there?
That's a great question.
Yeah.
The key difference is permanence and, crucially, independence from daily meds. So with a functional cure, yes, the virus might still be present in tiny amounts, hiding somewhere. But it's permanently controlled by the person's own immune system, without needing any daily antiretroviral therapy, or RT. The person achieves long-term remission. They suppress the virus, essentially, forever on their own.
Ah, okay. So no daily pills, no injections. The body handles it.
Exactly. And that's the goal that many, probably most, scientists see as more scientifically achievable, maybe in the nearer term.
So what's stopping that total removal? Why is the sterilizing cure so much harder?
This is really the core of the problem. HIV hides. Soon after infection, it integrates its genetic material right into the DNA of our own immune cells, specifically resting CD4 T cells.
It becomes part of our own cells.
Precisely. It takes refuge in what we call viral reservoirs. And our current medicines, RT, even though they're incredibly effective at stopping the virus from replicating in the blood.
Yeah, they're amazing now.
They are. But they just can't easily reach or clear out these hidden copies inside the resting cells. So if you can't completely flesh out that reservoir, you can't really claim a sterilizing cure.
Got it. So let's just state it plainly. As of today, right now, there is no FDA-approved cure for an existing HIV infection, full stop.
Correct.
We have excellent treatments, the RT you mentioned, which suppress the virus incredibly well. People live long, healthy lives. Transmission is prevented. But wait a second. If RT is so good now, healthy lives preventing transmission, Why is the FDA still insisting on a therapy that has to clear out this difficult reservoir? Isn't a durable functional cure where the body controls it without meds good enough for approval right now?
That touches on a really crucial point about regulatory thinking. It's about the standard of care and risk assessment. RT is amazing, truly transformative, but
There's always a but.
there is. It requires daily, lifelong adherence. If you stop RT, the virus rebounds, usually pretty quickly, precisely because it's hiding in those reservoirs.
Right.
So a cure, by the FDA's definition, has to fundamentally solve that problem. It needs to deal with the reservoir in a way that doesn't require constant, lifelong drug intervention. Think of it like RT manages the fire really well, keeps it contained. A cure needs to permanently eliminate the fuel source so the fire can't restart.
Okay, that makes sense. It's about getting off the lifelong treatment treadmill.
Exactly.
But, all right, while that cure timeline might still feel a bit distant, the progress in other areas like prevention and diagnosis, that speed has been pretty stunning lately. Our sources highlighted some big recent wins.
Oh, absolutely. The momentum there is undeniable. Just think about the FDA approval back in June 2025 for lenacapavir. You might know it as Yeztugo.
Right, the injection.
Yeah, the twice-yearly injection. That's a huge shift for prevention, for pre-AP, pre-exposure prophylaxis.
It's kind of incredible. I mean, just 5 years ago, the daily pre-EP pill felt revolutionary. Now we're talking about an injection only twice a year. That pace, it must put enormous pressure on the whole medical system to adapt.
It does. And the technical detail there is pretty neat, too. Lenacapavir is what's called a first-in-class capsid inhibitor.
Capsid. That's the virus's protective shell.
Exactly. It's like the virus's little protein coat. By inhibiting that capsid, the drug actually disrupts several different really critical stages of the HIV life cycle. And because it works differently from older drugs, and crucially stays in the body for a very long time.
That's how you get the twice-a-year dosing.
That's how. It completely changes the game for patient adherence, for convenience. It's a major step forward in prevention.
And the other big recent win you mentioned was on the testing front, right? Making it faster, easier.
Yes, the approval of that 1-minute at-home HIV self-test in STI. Getting a result that quickly in private, it lowers the barrier to testing significantly.
Makes it less intimidating.
Much less. And this really matters because, while we're all hopeful and working towards a cure, the most powerful things anyone can do today are get tested early, use pre-AP if you're negative and at risk, and stay consistently on ART if you're positive.
So don't wait for the miracle cure to take charge of your health now.
Absolutely not. Use the amazing tools we already have.
Okay, that's a great synthesis. So we've established the big scientific hurdle is that darn reservoir. But the path from having a cool scientific idea about tackling the reservoir to actually getting an FDA-approved therapy, well, it seems to break down into two major kinds of roadblocks. The biology itself, and then the regulatory hurdles.
Precisely. Let's tackle biology first, the hiding virus. As we said, HIV stitches its genetic code right into the DNA of our resting immune cells, those CD4 cells.
The time capsules.
Kind of like viral time capsules, yeah. So any cure therapy has to do one of two incredibly difficult things. Either find and somehow destroy every single one of those latently infected cells, or figure out a way to permanently silence that integrated HIV DNA so it never wakes up again.
And the difficulty there is
It's scale and location. You're essentially looking for a needle, or maybe millions of needles, in countless haystacks spread throughout the entire body. Not just in the blood, but deep in tissues like the gut, lymph nodes, maybe even the brain, places where drugs don't easily reach.
wow. Okay, that sounds daunting.
Yeah.
And if you're developing really powerful therapies to try and hunt down those hidden cells, that must lead straight into the regulatory challenges, right? Safety has to be paramount.
It does, absolutely. If we're talking about things like gene editing like CRISPR, or fundamentally reprogramming someone's immune system, these can be irreversible changes.
Right.
And the FDA's primary mandate is public safety, protecting a broad population.
Right.
So the first huge regulatory hurdle is safety and side effects. Any potential cure must be proven safe across diverse groups of people.
Yeah.
And the risk has to be considered acceptable for what is now, thanks to ART, generally a manageable chronic condition, not an immediate death sentence.
That risk-benefit calculation changes when the current treatments are so good.
Exactly. And the final, really time-consuming regulatory piece is durability. Proof over time. You can't just show the virus is undetectable for a few weeks after stopping RT and declare victory.
No, that wouldn't be convincing.
Not at all. This is the part that really slows the whole process down. The FDA requires solid proof that the virus does not rebound for a long, predefined period. Often we're talking several years after the person has stopped their standard RT.
Years without meds and without the virus coming back.
Correct. And running those long-term studies. They're slow, they're incredibly expensive, and they are absolutely essential to prove you have a genuine, durable cure, not just a temporary blip.
Okay, so with those biological and regulatory mountains to climb setting the stage, let's finally turn to the labs. What are the main strategies, the sort of four big approaches researchers are pursuing right now to try and crack this reservoir problem and achieve that durability?
Right? These are kind of the heavy hitters in cure research currently. The first major area is gene editing.
Okay, that includes tools like CRISPR, which people hear a lot about. How are they trying to use that, like cellular level precision surgery against HIV?
Well, the idea is precision surgery. There are two main ways they're trying this. One is to literally try and cut the integrated HIV DNA out of the infected cell's genomes.
Snip it out.
Sniff it out. The other approach, which might be a bit safer and maybe more achievable, is to modify a person's uninfected immune cells, make genetic changes, so that HIV simply can't infect them in the first place, like making the door permanently locked to the virus.
Okay. And where are we with that? I know there have been some early trials like EBT 101.
Yeah, early trials have moved forward. And the good news is they've generally shown these approaches can be safe, at least in the short term. That's a crucial first step. But the big issue so far is efficacy. It hasn't been completely effective, meaning the virus still rebounded, it came back, in some participants once they stopped their RT. The core challenge seems to be delivery and completeness. These editing tools might only successfully edit a fraction of the necessary cells, maybe not enough to control the whole reservoir. Or they might struggle to reach those deep tissue reservoirs we talked about.
Still working out the kinks. Okay. So, if gene editing is the surgical approach, the next strategy sounds different. Broadly neutralizing antibodies, or BNABs. That sounds more like deploying a specialized pharmaceutical strike force.
That's a good way to think about it.
Yeah.
BNABs are antibodies, but they're special ones, often discovered in certain people who naturally control HIV well, and then engineered in the lab. Their power comes from their structure. They're designed to target parts of HIV's outer surface, its envelope protein, that don't change much across many different strains of the virus.
So they can hit lots of different HIV variants.
Exactly. That's the broadly part. They can neutralize a wide range of HIV strains.
And how does that help towards a cure, beyond just maybe preventing infection?
Well, the idea for cure is twofold. One, they can help block any remaining virus from infecting new cells. Two, they can sort of flag infected cells for destruction by the patient's own immune system. They act like enhancers for the immune response.
And the status?
They're definitely promising, especially when used in combinations, maybe to help control the virus during periods when someone stops ART under medical supervision, what we call structured treatment interruptions. But as a standalone cure therapy, they still seem to struggle to maintain that control long term, likely because, again, they don't fully clear out those deep, latent reservoir cells effectively enough to meet that crucial durability requirement.
Okay, hitting that reservoir is tough.
Mm-hmm.
Which brings us to the third strategy, cell therapies, and specifically CAR-T therapy. Now, this is where we're actually taking out a person's own immune cells and reprogramming them, right? Using something called a chimeric antigen receptor.
Exactly. CAR-T has been a huge deal in cancer treatment, and the idea is being adapted for HIV. You take a patient's T cells, their main immune fighter cells, out of their body. Then in the lab, you genetically modify them to express this special receptor, the CUR. This CR is designed to specifically recognize proteins from HIV on the surface of infected cells. Then you grow millions of these engineered cells and infuse them back into the patient.
So you're basically creating a personalized army of HIV hunter cells.
That's the concept, guided missiles for HIV-infected cells. It's designed to overcome the ways HIV usually evades the natural immune system.
Sounds incredibly powerful, a really directed attack on the reservoir, but it also sounds really complex, intensive, heavy manipulation of the immune system.
It is. It's a potent idea, no doubt. But because you are fundamentally altering the immune system so significantly, the FDA's bar for proving long-term safety, making sure these engineered cells work persistently without causing serious unintended immune side effects, maybe even decades later, that bar is extremely high.
So still early days for HIV CAR-T.
Relatively early, yes. Clinical trials are ongoing, but they need years, really, to demonstrate both durable efficacy, that they keep the virus suppressed long-term off RT, and that crucial long-term safety profile.
Right. Okay, and that leads us to the fourth main strategy, the one that seems to tackle the hiding problem most directly. the kick and kill approach.
Yeah, the name is pretty descriptive. The theory behind it is actually quite elegant, since the main problem is HIV hiding silently inside cells.
You need to wake it up.
Exactly. That's the kick. You use drugs called latency reversal agents, or LRAs, to essentially wake up the dormant HIV DNA inside those resting cells, force it to start making viral proteins.
Making the hidden cells visible again.
Precisely. Once those cells are active and showing HIV proteins, they become visible targets for the kill phase. The kill could be enhanced art, maybe those BNAbs we talked about, or ideally the person's own immune system, perhaps boosted in some way.
Wake it up, then take it out. Makes sense in theory, but I know the results here have been kind of mixed, right? What's not quite working as hoped in the trials.
Yeah, mixed is a good word. We seem to run into two main issues. First, the kick agents often aren't specific enough or potent enough at safe doses. To wake up all the hidden virus across all those different reservoirs, you might need doses that cause too many side effects, too much systemic toxicity for the patient.
So the kick isn't clean enough.
Right. And second, even when some virus is successfully kicked out of latency, the subsequent kill mechanism often isn't efficient enough. It doesn't eliminate those newly activated virus-producing cells quickly or completely enough before they maybe start spreading the virus again, or just overwhelm the killing capacity.
So you wake some of it up, but you can't clear it all out reliably.
That seems to be the challenge. We can show parts of it work in the lab, or even in patients we can detect virus being kicked out. But getting the combination to work reliably, effectively, and safely across diverse groups of people to the point where they can stop IRT permanently, we're not there yet.
Okay, wow. This review of the different scientific angles, it really does make that regulatory timeline seem much, much clearer, especially when you factor in that demand for long-term proof, that durability data the FDA needs, showing it works for years, not just weeks or months.
Absolutely. If a potential cure candidate has to prove, let's say, 3 to 5 years of durable remission after stopping RT, and that has to be shown in large, diverse groups of people in phase 3 trials, you can immediately see why this isn't a fast process.
Right. It just takes time to gather that evidence. So let's try and put some realistic timeframes on this based on everything we've discussed. What about the next 1 to 3 years? Any chance of an approved cure then?
Highly unlikely for a widely available FDA-approved cure. What we will see, almost certainly, is more progress reports from ongoing trials, maybe some trials expanding, perhaps entirely new targets or combinations being tested in early phases. But an actual approved cure for the general population? No. We're still very much in the refinement and testing phase for all these core technologies.
Okay, reality check there. So, pushing out a bit further, what about the 3 to 7 year horizon? Is that more plausible?
This time frame.
Yeah.
This is probably the most optimistic yet potentially realistic window for a functional cure to emerge.
Okay.
If one of these strategies, maybe more likely a combination approach, perhaps linking a really effective kick with something like enhanced cell therapy or potent BNAbs, if that can successfully demonstrate long-lasting remission without daily meds in large late-stage phase 3 trials, then yes, we could potentially see a regulatory filing and maybe even an approval within that 3 to 7 year window.
So the first success is likely to be functional, not sterilizing.
That's the strong consensus, yes. That's the most probable first major breakthrough in terms of an approved therapy.
And then the ultimate dream, the sterilizing cure, getting rid of every last bit of virus.
That remains the toughest scientific nut to crack. Given the sheer biological complexity of finding and eliminating every single latently infected cell in every reservoir throughout the body, that path is likely much longer. We're probably looking at 7 to 10 years out, maybe even longer, before that becomes a realistic possibility.
A decade or more.
Could be.
Yeah.
But it's important to remember, the path to a sterilizing cure will almost certainly run directly through achieving and understanding a functional cure first. It allows us to test and refine these powerful technologies incrementally, safely.
Okay. That makes a lot of sense, which brings us right back around to what people can and maybe should focus on today. This has been a fascinating, complex, and I think ultimately hopeful deep dive into the future. But the knowledge we've gone through, a lot of it is really actionable right now.
Absolutely. That's critical. While everyone is understandably eager for these major cure breakthroughs, the strategies we have available today are incredibly powerful and frankly underutilized by some. You should definitely be leveraging the advancements we already have.
Okay, so let's list them out. Practical steps for listeners. First, get tested, right? Use those faster, more private options we mentioned, like the 1-minute self-test, or go to a clinic or lab regularly if you might be at risk. Knowing your status early is key.
Absolutely key. Early diagnosis means earlier access to treatment if needed, which leads to much better long-term health outcomes. Second, prevention. If you're HIV negative but might be at risk, talk to your doctor or clinician about pre-P.
And remember those long-acting options.
Yes. Don't forget, options like that twice-yearly injection are available now. They can be game changers for making prevention easier to stick with.
Okay. Third, for people who are living with HIV, treatment. Stay on your modern ART-key regimens. They work incredibly well.
Exceptionally well. They keep you healthy, allow you to live a normal lifespan, and crucially, when your viral load is undetectable thanks to RT, you cannot transmit the virus to others. Treatment is prevention.
Undetectable equals untransmittable. US.
It's exactly you over shoe. And finally, we talk quite a bit about those cure trials. If you're considering joining one, please only do so with really good medical advice from your own doctor. Understand what's involved. Clinical trials are absolutely vital for scientific progress. We need them. But they always carry potential risks. They require a significant time commitment, intense monitoring. Be fully informed about the specific trial, the potential benefits, and the potential hazards before you enroll.
Sound advice. You know, stepping back, despite the long timelines for a cure, the progress really does feel tangible.
It absolutely is. The pace of research, the sophistication of the tools being developed now, it's genuinely much faster and more advanced than even a decade ago. And every success we have, whether it's in prevention like new PIAP options, easier testing, or incremental gains in understanding the reservoir, it all helped reduce new infections and moves us closer to solving the remaining big challenges for a cure.
So, the path to that FDA-approved cure when it comes will likely be gradual. It probably won't be one single magic bullet, but maybe smarter combinations of therapies we're developing now.
That seems most likely. Combinations targeting different aspects of the problem. And the absolute key, the metric everyone will be watching, is durability. Proving that control, that remission off RT, lasts for years, not just months.
Right. So we'll leave you, our listeners, with maybe one final thought to mull over. The ultimate success and impact of a cure, whenever it finally arrives, actually depends quite heavily on how well we use the tools we have today. How many people get tested, start PrEP if needed, get onto and stay on effective RT if positive.
That foundation is critical.
It really is.
Yeah.
So testing early, staying informed, accessing modern prevention and treatment, that's genuinely the most powerful action you can take right now for your own health and for the community. Don't wait to know your status. And please, consult reliable public health resources. We'll link some to find verified testing centers in your area today.